Okay, uh, so last time we were talking about the process of cleavage and how from the zygote you go from a diploid cell to the two-cell stage, to the four-cell stage, 8, 16, which is the morula, 32, etc. And then you reach the point where at day 6 you form a hollow sphere that it has some cells that are called the trophoblast, and in the one of the poles or one of the sides of this cell, you have the inner cell mass, which is going to form the embryo. And then at this stage, this structure is called the blastocyst. And in between the inner cell mass and the trophoblast, you have this cavity that's called the blastoquele, and in here you have this fluid that it fills this cavity. So the inner cell mass is going to form the embryo, and this is also known as embryoblast. Anytime that you hear in biology the ending blast, that means it's mature. And then the embryo proper then forms from this embryoblast. And then you will have the implantation of this blastocyst in the superior posterior part of the uterus, typically in day 7. And then if the cells separate, you can have twinning. So this will be what is called monozygotic or identical twins. This is different from dizygotic or fraternal twins, which form from two different zygotes. That's when two egg cells will be fertilized each by one sperm cell. Identical twins, basically, they share more information than dizygotic or fraternal twins, and they look more alike than the dizygotic twins. And this happens because this mass of cells can split. And they're supposed to split within a certain period of time, because if they don't split, you will have what's called co-joint twins. So this is the different days. And every time that I have a figure, I have to come back here and point. So day 1, you have your secondary oocyte, where the sperm cells come, erode the coronary radiata. The one that reaches this zona pellucida will bind to the zona pellucida receptors, and then it will penetrate this zona pellucida, bringing the head of the sperm cells inside into this cytoplasm of the egg cell, and then forming with the nucleus, specifically the part of this head that, which is the nucleus, it will form a pronucleus. And remember, polyspermy, it is prevented, or the fertilization by more than one sperm cell, by shutting down these receptors in the zona pellucida. So this is part of the cortical reaction. So these two nuclei, the one nucleus, from the one from the paternal side, and of course the maternal side in the egg cell, they get together, and then they form a single nucleus, which is going to be the what is called the amphimixis. So you will complement now, since you had 23 chromosomes in each gamete, you complement the diploidy, which means that you have to have 46 chromosomes. And now this structure now is going to be called the zygote. And look at how it happens all in this region. So fertilization happens between the ampulla and the isthmus, or the infundibulum, sorry. And then here you have the zygote, so it will migrate. The zygote will migrate towards the uterus, and then it cleaves into two blastomeres That's the 2-cell stage. And then this will cleave in half or split in half, so you form the 4-cell stage, 8-cell stage, early morula, and then you form in day 4 the blastocyst, and then between days 4 and 7, but actually it's in day 7, that's where you have the implantation, and then here you have these trophoblast cells, the blastoquel with this fluid, and then here the inner cell mass. Now, this inner cell mass, which is going to form the future baby, it will be kind of basically facing the myometrium and the endometrium. It doesn't face the uterine cavity. Why do you think is that? Why this inner cell mass orient to go towards the myometrium? What is so special about the myometrium? Why do not orient toward the cavity? Sometimes they send emails to people saying, well, there's free pizza. What does orient you to where the free pizza is if you don't know the building and the room where that pizza is? What it makes you walk towards that area where the pizza is? The smell. So what mom has in here? Nutrients. The glycogen. This area, it's rich in glycogen, so the baby follows where the nutrients are. So that's why the inner cell mass orients towards this direction. So now, once this blastocyst, in day 7, binds into the endometrium, it will start secreting digestive enzymes to start degrading this endometrium. So the trophoblasts after are going to start dividing into a cellular trophoblast or cytotrophoblast. That's why the name is cyto, because cyto means cell. That is the internal trophoblast, while the outer syncytial trophoblast, which is basically a multinucleated cell, is large, is going to be non-antigenic, and will not trigger an immune reaction from the maternal tissue. So then in the inner cell mass, you will have cells that differentiate into 2 layers, the hypoblast and the epiblast. And they form a disc that is going to be called the embryonic disc. So the hypoblast cells are going to be below, that's why it's hypo, and the epiblast cells are going to be above. The epiblast cells are going to be taller than the hypoblast cells, so they're going to be columnar. And then out of these 2 layers, the hypoblast and the epiblast, you will form the 3 germ layers. So that happens between day 7 and 14. By the end of the 2nd week, you will start forming these 3 layers. So during the 2nd week, you have 2 layers, the hypoblast and the epiblast cells. And then starting on week 3, you have the 3 layers, which are the 3 embryonic layers that we call endoderm, mesoderm, and ectoderm. So 2 weeks, 2 layers, 3 weeks, 3 layers. And how does this happen? Well, it happens through a process that is known as gastrulation, in which part of these epiblast cells will migrate in between the hypoblast and the epiblast cells, and then we form a middle layer, which is going to be the mesoderm. Now, something that is happening in the trophoblast, because that change happens in the embryo, so you form out of the 2 layers the 3 layers, but in these trophoblast cells you will have a change in which these trophoblast cells, the syncytial trophoblast, will start migrating deeper into the myometrium, like burying itself inside the wall of the uterus, and then they will follow chemical signals that are found in the blood vessels. It is following actually the blood vessels, this syncytial trophoblast. And then with these digestive enzymes that these cells have, they will form intervillous spaces around the blood vessel that are known as lacunae. And then why does this happen? Well, because in the blood vessels there is oxygen, and there are nutrients that the mom provides for the baby. So it's another example of chemotaxis, and that happens by day 12. And then by day 16, there is a complete invasion. The uterine wall will be embedded and completely covering the blastocyst. So this is the blastocyst here, superior, posterior part of the uterine wall, hopefully, that's where it implants. This is the endometrium, this is called stratum functionalis. And then the inner cell mass again faces the uterine wall, so you magnify this part. This is day 7. And then these inner layers, this specifically in this region, this will be the cytotrophoblast. It's near to the inner cell mass. And then the external layer that starts growing, that forms a large mass of cells, this will be the syncytial trophoblast. And these are glands within this endometrium. This gland secretes glycogen, so attracts this blastocyst, and then the blastocyst contact this stratum functionalis, starts extending this syncytial trophoblast. See how by day 8, this syncytial trophoblast is deeper within this uterine wall. And then you start forming these 2 layers of cells, the hypoblast and the epiblast. And then by day 12, this syncytial trophoblast goes deeper, following these blood vessels. They like oxygen. Oxygen is in here, plus the nutrients that the mom gives. So they start forming these lacunae. And then you start forming the amniotic cavity, and then you will have, by day 16, a completely buried blastocyst. So it has invaded already the maternal wall of the uterus, and it starts finding as many blood vessels as it can, and also the uterine glands that gives glycogen. And these cells of this syncytial trophoblast doesn't have antigens, because if they have antigens, then the immune system of the mom will destroy this. It's a temporary implantation. And this new person will have different antigens from the mom. So the mom has to embrace and protect the baby by not attacking its cells, and it will be a physical and a chemical protection. This syncytial trophoblast cell secretes this hormone that is called ACG, human chorionic gonadotrophin. This is the one that gets detected really early in pregnancy. And this HCG stimulates the corpus luteum in the ovaries of the mom to secrete estrogens and progesterone. Estrogens are growth factors, and progesterone helps for the development of the placenta. And then HCG, it can be found in urine, but its levels increases very high in the blood, and then you can have detection of pregnancy with this pregnancy test. And then if there is a genetic defect in the baby, that will lead to a spontaneous abortion or miscarriage. It's only until you find out, if you grab part of the tissue, and then you find what defect it was, but most spontaneous abortions happens because there is a genetic defect that it is incompatible with life. And then if the blastocyst implants in other region, if it's outside of the uterus, this will be called ectopic pregnancy. Ectopic pregnancy can happen due to several reasons. And this ectopic pregnancy happens between 1 to 2% in the uterine horns. These are called tubal pregnancies. When you have these pregnancies, you have the risk of the rupture, of course, of the fallopian tubes, because that's not wide enough to allow the growth of the baby there. And that can lead to bleeding and death of the mother if she doesn't receive proper attention. So other places where you have ectopic pregnancy can be the abdominal cavity, ovaries, or the cervix. Kind of crazy how it can happen in the ovaries. Imagine a sperm cell jumping to try to find the ovaries. And then if you don't have medical intervention, then that can lead to maternal death. Now, early in development, you also have to form these extraembryonic membranes that appears first during the second week of development from the embryonic disc and the trophoblast. So these membranes helps to bring nutrients, gas, and then remove wastes from the baby. That includes the yolk sac that eventually will become part of the digestive system. And in this case, the yolk sac as well start forming red blood cells very early. And then you have another membrane that is called the amnion, the allantoids, and the chorionic villi. And the chorionic villi, something that it helps is to increase the surface area so that there is connection between this chorionic villi and the endometrium so that you can have greater exchange of nutrients and oxygen by diffusion. So before I continue, let me tell you that there's actually no physical connection between the maternal blood vessels and the fetal blood vessels, never. Why? Because chances are that the blood of the baby is different from the mother's, and then you don't want to have destruction of this blood from the baby. So then the part of the baby that it is close to this syncytial trophoblast is going to form an extension that is called the yolk sac, and then you start forming as well the umbilical cord that is going to be having this single vein, which is the umbilical vein, and the paired umbilical arteries. So you will have then the umbilical arteries that carry away CO2-rich blood from the baby, and then the CO2 diffuses into the maternal blood vessels, and then oxygen is delivered into the baby by this umbilical base. And then this will be the amnion, sorry, this one, and this will be the amniotic cavity, and here you have the embryo. And then this is the yolk sac. So the yolk sac specifically arises from the hypoplast, and it's going to be again forming part of the digestive system, and it's going to start forming early red blood cells in the baby. And then it's going to as well form the germ cells or precursors of gametes that eventually will go into the gonadal ridges that are going to form part of the digestive system, reproductive system. The amnion arises from a cavity in the epiblast. And then the fetus or the conceptus is going to be surrounded by amniotic fluid, and this amniotic fluid is going to allow the growth of the baby, and it's going to come, this fluid is going to come from the mother's blood and later from the embryo urine. And this amniotic cavity is going to be penetrated only by the umbilical cord. The allantoids is going to be part of the posterior wall of the yolk sac. That develops during the 3rd week. And this is going to lead to the formation of early blood vessels. And then it becomes the umbilical vessels, these allantoids. And then you have a duct that is going to run to the urinary bladder, which it drains it. And then you also will have the formation of the chorion that develops from the cytotrophoblast and the syncytial trophoblast. And this one is going to extend some chorionic villi that looks like little fingers that forms entirely the chorion. And then eventually this will grow towards this stratum basalis layer in the endometrium, and it's going to be forming part of the placenta later. So here is the summary of the 4 extraembryonic membranes and their functions. So yolk sac is going to form part of the digestive system, forming red blood cells, blood vessels, and germ cells. The amnion is going to protect the baby. It's going to hold this amniotic fluid and it's going to produce it. And that helps to protect the baby physically from trauma, also helps to maintain the body temperature in the baby, and it helps also for the symmetrical development of muscles, and then prevents the drying out and adhesion of the body parts of the baby during growth. Allantoids is going to form the base for the umbilical cord, and it's going to link the conceptus to the placenta. And then later on, it's going to form the urinary bladder. Chorion is going to enclose all the extraembryonic membranes. It's going to form the chorionic villi that is going to help for the absorption of nutrients. So details about the formation of these germ layers. So in gastrulation, you reorganize the cells and you form this trilaminar disc in week 3. As this happens, you will form in this disc, in the middle, a little indentation or groove that is going to be called the primitive disc, and this is going to be in the dorsal surface of the epiblast. This disc, this primitive streak is going to extend from head to tail regions, from caudal to cephalic regions, and then it's going to establish the left and the right side of the embryo and the dorsal and the ventral surface. And then the gastrulation begins with the ingression, so where some cells move from the epiblast underneath the primitive streak, and then they will form the mesoderm. So the endoderm is going to come from the hypoblast, so you will replace it. The mesoderm, again, will be those cells that migrates between the epiblast and the hypoblast. And then the ectoderm will going to become, we're going to be part of the, or well, they come from the epiblast cells. So why we call them endoderm, mesoderm, and ectoderm? Well, endo because it's going to be internal, mesoderm because it's going to be in the middle, ectoderm is going to be external. So if you make a section of this part of the three germ layers, so if you see it, so you will see that you have a cephalic end, a caudal end, and then you form this primitive streak that is going to form the left and the right side. This is going to be the pelvic area, this is going to be the head. And this is the amnion. So if you make a cut and then you see this bottom part, you can see here the epiblast, which are these simple columnar epithelium, and these hypoblast cells, which are basically cuboidal cells. And then as this primitive streak moves towards both ends of the embryo, from the caudal end into the cephalic, you will see the ingression. So basically part of these endings or edges, from this primitive streak, part of these cells will migrate of the epiblast, and then they will form the mesoderm. And then you will end up by week 3 with the ectoderm, which was previously the epiblast, the endoderm, which was previously the hypoblast, and the mesoderm, which were these cells that basically enter in between the two layers. So then you end up with ectoderm outside, mesoderm in the middle, and endoderm inside. As this happens, there is going to be folding internally. Like you will have this. The cells will make this migration, and then you will form a tube. So by week 4, the embryonic disc will become cylindrical. And then you will form a folding that is called cephalocaudal folding, in which the baby is going to fold in like this. And then you will have a transverse lateral folding that is going to be the left and the right side of the embryo. And then as this happens, you will form the future trunk region and then the primitive gut. That will become, of course, the digestive system. And then as this is happening, you start pinching the yolk sac, and then you start forming the betaline duct out of the endoderm. So this is the cephalocaudal folding. So this is going to be the future umbilical cord. This is the allantoids. This is the yolk sac. And then the baby keeps folding. It keeps constricting this yolk sac. And then you start forming in late week 4 the heart. And this is going to be the gut, the primitive gut, with a cephalic end and a caudal end. So now, if you look from side to side, so this is left, this is right. So here you have a mesoderm, the ectoderm, the amniotic cavity, and the yolk sac. So you start constricting the yolk sac, and this is going to be forming the primitive gut. Here on the sides, you will form the thoracic cavity, the abdominal cavity, and this tube, which it comes from the ectoderm, is going to be the future spinal cord. So this is the neural tube, and as well is going to be the future brain. So we have a tissue in the embryo. It's called the mesenchyme. And this mesenchyme is going to be what is going to form all of these cells, the epiblast, hypoblast, mesoderm. And then out of these three layers, we will form the different organs. Most of the organs have derivatives of each of these three layers. And the formation of organs is called organogenesis. So when organogenesis begins, the embryo is about 1.5 millimeters or 0.6 inches in length. So very, very tiny. And then the embryonic period ends at the end of week 8, and the embryo measures about 1 inch in length. So by this time, you have recognizable organ systems, and they will start functioning. But not all the organs will be functional. For instance, the lungs, they need to have a maturation process that happens towards the end of pregnancy. Now, what are some of the derivatives of the ectoderm? So the ectoderm is going to form the epidermis in the skin, which is the most superficial layer for skin. And then the nervous system will happen through neurolesion, which is basically the process of forming organs in the nervous system. So we have first, next to this primitive streak, this neural plate. And this neural plate is going to fall inward, it's going to deepen, and it's going to form the neural tube. Then it's going to pinch off from the ectodermal layer, and then it's going to form this complete cylinder by the end of week 8. And then on the cephalic end, you will have some dilation, which is going to be called the primary brain vesicles, that are going to form the forebrain, midbrain, and the hindbrain, that are going to form the different parts of the brain. Now, on the sides of the neural tube, we will have a separate clusters of cells that are going to be called the neural crest cells. So these ones, they migrate into different regions, and they form the cranial, the spinal, and sympathetic ganglia and associated nerves. And in our skin, they're going to form the melanocytes. And also, they're going to migrate towards the middle of the adrenal gland, forming the enterochromaffin cells, and they are also going to form connective tissue cells. Sometimes we have absence of migration of these cells, of these neural crest cells into our skin. And then we end up with little patches of skin that are not colored. So that's the information about the ectoderm. Now about the mesoderm. So for the mesoderm, you have cells that are immediately deep to this primitive streak that forms from this epiblast migrating in between the epiblast and the hypoblast. And they form a structure that is going to be called the notochord. And that is going to organize the embryo around a central axis. And then on each side of the central notochord, which by the way, the notochord is going to be a source of signals for development, for bilateral development. So on either side of this central notochord, you will form a cluster of cells that are going to be called the somites. The somates are going to have three regions. The sclerotome, that is going to form the vertebrae in the ribs. Dermatome, that is going to form the dermis in our skin. And the myotome, that is going to form most skeletal muscles. The sclero, which is the most hard tissue, that's why it's called sclero, is going to form bone, basically. Dermatome, the dermis, and myotome, muscles. Myo, remembers, means muscles. And then we have lateral to these somites the intermediate mesoderm that is going to form the gonads and then the kidneys. And out of a sheet that is called the lateral plate mesoderm, you will form organs like the spleen, the adrenal cortex, most of these cardiovascular system organs, and then the serous membranes of the body cavities, and the connective tissue that makes our limbs. So we have then these other derivatives of the mesoderm. For the endoderm will form the epithelial layers of the internal organs like the digestive tract, respiratory tract, urinary tract, and reproductive systems. Basically, all the mucosas internally. That's the endoderm. And then, of course, part of the epithelia from all of these systems, we have the derivatives of this epithelia, which will be the glands and accessory digestive organs. And also some endocrine glands like the thyroid, parathyroid, thymus, palatine tonsils, most of the liver, the gallbladder, and the pancreas. And that's the summary of all of these derivatives. So, here we have an MRI, mini MRI of the embryo at week 4. This is a lateral view. So this is the caudal end, this is the cephalic end. So in the thoracic area, you can see the protrusion of the heart. And then in here we have the arm and the leg buds. These are the somites. This is on the sides of the neural tube that is going to form the spinal cord. And then in here we have the branchial apparatus that is going to form several structures in the neck and in the face. And here's the optic placode that is going to form the lens. This is the eye. This is the forebrain, hindbrain in here, and this is the ear. And then by week 8, most of the organs are formed and mature. You have the umbilical cord. This is the amniotic cavity, chorion villi. Look at the blood vessels in the brain, and the eye, the ear, and completely formation of these limbs. It's an amazing transformation only within 4 weeks and 8 in total for organogenesis. So this is the most critical period of development because that's when we are forming our organs. And anything that can damage our organs during that life, it is a teratogen. So it's a substance or a physical agent that can cause congenital disorders or even death. So some of the infectious agents that can damage the baby will be viruses like rubella, bacteria, also some protozoans like toxoplasma. HIV as well can cause problems in the baby. Substances like alcohol, nicotine, prescription drugs, aspirin, anticoagulants, and some antibiotics are teratogenic. in the 1960s, some women took some pills that contained thalidomide. This was a drug that it was used to prevent morning sickness. However, this one created problems because the babies couldn't develop the upper limb or the lower limbs, so they end up with short limbs. So that's organogenesis. Now let's talk about the formation of the placenta. So the placenta is a disc-shaped organ that attaches the baby umbilical cord into the uterine wall. And this is an organ that helps for temporarily exchange oxygen, nutrients, and wastes. As well, it produces hormones like relaxin, HCG, progesterone, and estrogens. The ovaries will be able to sustain the pregnancy for a short period of time with the corpus luteum and the estrogens that they secrete. But then the placenta, since it's larger, can produce larger amounts of these hormones, and then it will take over the function of the ovaries with time. So during pre-embryonic and embryonic periods, the conceptus receives nutrients from the urine milk from the endometrial cells. So basically, it will receive this glycogen, for instance. But by week 12, since you form the placenta is going to be a major source of these hormones. And then that will sustain the pregnancy for the rest of the pregnancy. Now, the placenta can attach to different regions in the uterus, and one of these regions can be the cervix. Now, if the baby is not going to be born through C-section, if it's going to be born through the cervical canal, do you think it's a good idea to have the placenta implanted there in the cervix? No, because the baby head cannot pass through the placenta, so this is known as placenta previa. Most of the cases are not fatal, but that can lead to a spontaneous abortion or premature birth. But most cases end up with live births. And something that happens is that the uterus has to stretch more to accommodate the fetal and the placental growth, and the placenta may lead to hemorrhage or become infected. Placenta previa, if it's detected earlier, it will be monitored and typically end up in C-section. Now, the umbilical cord connects the center of the placenta to the umbilicus of the fetus or belly button, and it has two umbilical arteries and a single umbilical vein. This umbilical vein carry away the wastes of the baby, and the umbilical, carry away oxygen reach the vein from the umbilical cord into the rest of the tissues of the baby, and the arteries carry away the wastes and the oxygenated blood. So during fetal life, there is an opening in the heart between the right and the left side that is called the foramen valley that allows the transfer of blood from the right side into the left. Because the baby, it is basically found inside the amniotic cavity, floating within this amniotic fluid, and it's constantly aspirating this fluid, and then out of this, you don't have basically oxygen available. So then the lungs, they don't have air, they are filled with this fluid, and then the baby, there is no purpose for transferring from the right side of the heart blood into the lungs, because the lungs, they don't provide any oxygen. So one way to shunt the blood, it is to have an opening between the right atrium and the left atrium, which is the foraminal valley, so you don't send this blood into the lungs. And then the umbilical vein that carries oxygen-rich nutrients, oxygen-rich blood and nutrients, it will be the single source of oxygen for the baby. And then when the baby borns, and then we are outside in the world inhaling air for the first time, when we have our first breath, that opening between the right and the left atria closes, and it forms these fossa valleys, which is the remnant of the foraminal valley. Now, the vein and the arteries are embedded within this Wharton jelly that insulates and protects these umbilical vessels. So here is the formation of the placenta. So by week 12, you have this umbilical cord connected into this cushion of tissue that is called the placenta. And then if you magnify this, you can see these fetal blood vessels. So you have this single umbilical vein that carries oxygenated-rich blood, and then two umbilical arteries. And then here we have the chorion, which is this large space. And then here you have the maternal blood vessels. And then you diffuse oxygen from the maternal blood vessels into this umbilical vein. And then these umbilical arteries, that they form capillaries within this chorionic villi, they diffuse CO2 and wastes into these veins of this of the maternal side of the placenta. So the space that is filled with blood in this placenta is called the placental sinus. And you have within this placenta a layer that is called the decidua, or stratum functionalis. This is the region beneath the fetus. And then you have the decidua basalis, which is the region around the uterine cavity. So the one that is by the uterine wall is the basalis, and the one that it is by the uterine cavity is the decidua capsularis. So if you magnify one of these regions within this area of the placenta, so you will see that there is the placental barrier. This is the maternal basal lamina, and then in here you have cells. This is the blood vessels of the mom. This is the blood vessels of the baby. Oxygen diffuses where you have higher concentration of oxygen, which is the maternal side, into the fetal side. Nutrients as well diffuses in this case, and wastes, the baby's accumulating wastes and transfer it into the mom. So this is a cytotrophoblast. This is a syncytotrophoblast. So does the mom have the same hemoglobin type as the baby? No. The baby has fetal hemoglobin, which has different globin chains that makes the baby to steal the oxygen from the mom. It competes from the oxygen. So this fetal hemoglobin binds much the oxygen. Once the oxygen reaches this hemoglobin, it doesn't let it go. So this is because, again, the baby is in disadvantage. It's not found in an area where it can get oxygen freely as when we are born. So the blood flow through the placenta. So the maternal blood flows through the pseudobasalis layer, through the maternal arteries into the placental sinus. And then as this happens, you have circulation of blood in the fetus entering by these umbilical arteries into the capillaries in the chorionic villi. And then, just as I mentioned before, you have the fetal hemoglobin that has greater affinity than the maternal hemoglobin. Okay, so that is the details about the placenta. Now, for the fetal development. So after the embryonic period, the embryo can grow as large as 50 centimeters during this period, which is 19.7 from crown to heel length, or CHL. And then you can use another length, which is called crown to rump length, or CRL. And the average weight of a full-term fetus ranges from 3.2 to 3.5 kilograms, 7 to 8 pounds. And then when, even during childhood, you have the head, which is disproportionately larger compared to the thorax and also to the lower limbs, which are low or short, sorry. And as we grow, the lower limbs and the upper limbs grows, and then the head becomes smaller. So by month 3 of development, so you have growth in length, you have formation of these upper limbs, the head is large because the neurons are developing, and also that happens during the first 2 years of life. That's where you have more development of the neurons. And then you can clearly see the ears that are present, but they are not prominent. And then you start ossifying most of the bones that previously were made by cartilage, hyaline cartilage. And then the genitals are distinguishable as male or female, and approximately the crown to rump length is 9 centimeters by month 3, which is 3.5 inches. And by this time, you have movement of the baby. And the uterus is mostly at the length of the navel area of the female. Month 4, what happens? Well, you have growth of the baby that is very rapid. So you have the lengthening of the lower limbs, and then you start formation of your joints. Your kidneys are well formed. The digestive glands, like the accessory glands, are forming. You can hear the heartbeat with an aid of an stethoscope. And the baby doesn't like to hear loud noises, so it startles and turns away from not only loud sounds, but also bright lights. And then the crown to rump length is about 14 centimeters or 5.5 inches, something like this, by the end of the month, 4th month. Now, something that happens in month 5 is that you have a little bit of slow growth, but you have the formation of your hair on the head and the skin. And then that hair is going to be very fine, and it's going to be called lanugo. We shed this hair after we're born, and then sometimes we keep a coat of lipids that is called sebum or Bernice gaseosa. So it's like yellowish, and that protects us from over-soaking in this amniotic fluid. Something as well that happens during this month is the deposit of brown adipose tissue that helps to keep the baby warm. And then you have as well the contraction of the skeletal muscles of the baby. And the length now is going to be 19 centimeters or 7.5 inches. So more or less something like this. And you can see more or less separated these fingers. Before that, we have our fingers attached together by a membrane. And this membrane make it look like if it was a tad, okay, of frogs. So this membrane disappears by apoptosis. So we have cells that start dying, and then what is the remnant? This little flap of skin. The most preterm that the baby will born, the most likely that you can see that connection not only between the fingers but also between the toes. By month 6, the fetus gains significant weight. You form the eyelashes, the eyebrows, your eyelids are partially open, and then because you have spent more time in this amniotic fluid, the skin becomes wrinkled and sometimes translucent, and then your lungs begin to produce this substance by these pneumocytes too, that is the surfactant that prevents the collapsing of the lungs when we take our first breath. And then the length is going to be almost 10 inches, 23 centimeters by the end of 6 months. By month 7, your eyelids are completely open. Fat is added into the subcutaneous tissue, and the skin, of course, will be wrinkled and now become reddish. Usually, the baby starts turning upside down and start assuming the vertex position in which the head is pointing towards the cervical canal. And then something that happens in males is the descending of the testicles that forms originally in the abdominal cavity. They descend through this inguinal canal pulled by a ligament that is called a gubernaculum that shortens and then brings them into the scrotum. And then the length, it is around 28 centimeters or 11 inches, which will be kind of this. Months 8 and 9. So the neurons in the baby's nervous system start forming networks. The organs grow and develop. You have more production of the surfactant. Blood cells now start forming in the bone marrow, not in the yolk sac. And then the digestive and the respiratory system, they complete their development during 9 months. And then the fetal skin becomes less wrinkled. And then you start shedding sometimes lanugo, but many babies, they born with their lanugo. And the testicles complete their descent into the scrotum. And the length is 12 inches by the end of 8 months and 36 inches by the end of 9 months. And that can vary. This is just like an average. So about 266 days after fertilization, the baby is considered full term. And then is ready to born. And then you have the formation of nail. Any questions? Okay, so prematurity. So a premature is an infant that borns 3 weeks before full term, which is about 38 weeks. So there is some percentage of babies that typically born full preterm here in the United States is 12%. And the earlier the baby born, the greater chances that it would have to have problems, including IRDS, which stands for Infant Respiratory Distress Syndrome. So it is important that when there is a risk for having a preterm, to help for the baby to develop their own surfactant. And this is by giving shots of corticosteroids that helps for the baby to form these surfactants, because these surfactants prevents the surface tension that we have in the lungs, in the alveoli, for the alveoli to collapse. So when you put this oily secretion, this surfactant, then the baby can expand their lungs, but if not, the baby can die. Now, infants may have minimal suckling and swallowing reflexes, and after they're born, and their livers may not produce clotting factors or albumin. So that's also part of the problem with prematurity. So since the liver is immature and they don't produce clotting factors, chances are, as the baby's born is passing through the birth canal, the ventricles in their brain get damaged, and then they can have a hemorrhage in the brain, which is called intraventricular hemorrhage. And then the baby also can develop edema out of these problems, and the hypothalamus may not be able to maintain the body temperature. So the most common cause of infant death, it is premature birth. And fortunately, there are several advances in neonatal care that increases the survival rate. So 30% for infants born at 23 weeks, the chances increasing or survival almost twice just 1 week later. So from 30%, you can go up to 60% of survival just by one difference in 1 week. And then a baby that borns at 25 weeks has 75% of survival and 90% between 27 to 28 weeks. And that's just related to this factor of the lack of production of the surfactant. Unfortunately, many babies can undergo problems like cerebral palsy that can lead to intellectual deficiencies and problems with vision and hearing. So then I told you before about the baby being in this amniotic fluid and capable of oxygenating its own blood. So then the baby will have this shunt that prevents a lot of blood going to their lungs, but also they have other shunts, like for instance the ductus venosus. This one communicates these blood vessels from the liver into the vena cava. And this is necessary for filtering and store these nutrients that basically bring, these nutrients are brought by the umbilical vein into the liver. So you don't have complete perfusion of these organs, and then you have to bypass part of this blood. And then the foramen ovale, I already told you, connects the right and the left atria, and it closes once we born. And then we have a communication between the pulmonary trunk and the aorta, which is called the ductus arteriosus, that also escape the non-functional lungs. And once we're born, these ductus arteriosus become the ligamentum arteriosum, and then the foramen ovale become the fossa ovale, and you will have then the ductus venosus converting into the ligamentum venosum. Now, once we're born and they cut the umbilical cord, these umbilical arteries and the umbilical veins convert into remnants. Like, for instance, the umbilical arteries will become the medial umbilical ligament, just in the internal part of the abdominal cavity. And on the sides, we will have the remnants, the remnant of the umbilical vein that will become the ligamentum teres, that is going to actually be the round ligament of the liver. So those are the remnants of these structures within the umbilical cord. So this is then the blood circulation in the fetus and in the newborn. So here we have foramen ovale shunting blood from right to left. And then here we have the the ductus arteriosus shunting blood from the aorta into the pulmonary trunk, or vice versa, sorry. And then in here, we have this shunt between the liver and the inferior vena cava. And then we have a single umbilical vein that carries oxygenated blood, and then we have two umbilical arteries that carry away this deoxygenated blood, bringing it here into these sinuses in the placenta, so that by diffusion, the CO2-rich blood, the CO2 goes into the maternal side, so that the mom can get rid of the CO2 produced by the baby, while this oxygenated blood that diffuses from the mom into the fetal side is going to be brought into the different tissues by this umbilical vein. And then once we're born, this ductus venosus becomes the ligamentum venosum. This ductus arteriosum becomes the ligamentum arteriosum, and the foramen ovale become the fossa ovalis. So there is no more shunt between right and left. So the right side of the heart will carry CO2-rich blood, while the left, oxygen-rich blood. So then we went from the zygote into the fetus, and then into a newborn. So postnatal development. In the first trimester, I was carrying away. So now for maternal changes. So we talk about fetal changes, now for maternal changes. So in the first trimester of pregnancy, basically many women can develop morning sickness, fatigue, and other symptoms. Morning sickness develops because, not in all women, because the ACG stimulates the vomit center in the medulla oblongata. And then fatigue. Well, the baby's stealing nutrients from you. Not only carbohydrates, but also proteins, also ions to form its own bonds, like calcium. So it's very important, and vitamins, so it's very important that the female has proper nutrition at every stage of pregnancy, not only in the first trimester of pregnancy. Something that happens to the baby if the female doesn't get folic acid is that the neural tube is not going to close, and the baby can have something that's called spina bifida. It can be different types of spina bifida, one that is called spina bifida occulta, in which the baby will have a dimple, especially in the lumbar area, and you can have tiny hairs coming out from there. Another one, it will be into which the meninges can protrude, and another one will be even part of the spinal cord. Thankfully, there are surgeries that, if that happens, of course, better is prevention, but if that happens, there are surgeries that can correct the spina bifida. And babies, since they have full potential, their cells undergoing a lot of mitosis, they can heal really fast in comparison to adults. So, but again, this is something that it needs to be prevented rather than treated. And then for the second trimester, many women feel relief from morning sickness. They maybe get used to this ACG, or there is less sensitivity from the receptors. And many women, they start feeling the movements of the baby for the first time. And in the third trimester, since the baby starts growing a lot in size, so the female will have compression of the urinary bladder, and the baby settles in the lower position in the abdomen, and sometimes that the women experience symptoms related to that, like backaches, or sometimes pain within this area, pelvic area. So we will leave it up to there. Any questions?